All right, for Bio 201, this is chapters 2 and 3, Bio 111 Review, Part C. This will be the third and final video on this. And in this one, we're going to finish out the remaining things in Chapter 3 for the Bio 111 review. So, Chapter 3 begins with a discussion of the cell and cell structure. And it starts on page 66. And going into 67 and so forth, the order of which doesn't really make too much of a difference. However, if you look on page 68 and 69, there is a two-page overview of all of the structures and organelles of the cell that you are going to be responsible for, at least to remember, from Bio 111. Obviously, there's no plant cells, so you're not worried about chloroplasts or a cell wall. This is a typical animal cell, has another cutout view on page 68, and then on the side and all the way on 69, it highlights the key organelles and structures that you need to be familiar with. The only thing in this whole thing here that you don't have to worry about are proteosomes. That's on 69, the left-hand side, second one down. You can forget about those, but everything else that is here you need to be familiar with, and all of this you should have had in Bio 111. And in the following pages, it goes through a lot of these organelles and structures and talks about their functions. Not in excessive detail. It's really no further than what you learned in Bio 111. Very quickly, the nucleus is the control center of the cell, contains the genetic material. We have the endomembrane system, which consists of the rough ER, the smooth ER, and the Golgi apparatus. Rough ER, protein synthesis because of attached ribosomes. There are also free ribosomes throughout any cell. Nothing says that ribosomes must be attached to the ER, but some of them are. Smooth ER, lipid synthesis, carbohydrate synthesis, storage, a whole bunch of different functions. Golgi apparatus is packaging, processing, and shipping of proteins. Vesicles transport things within the cell, can also have things leave the cell, or vesicles form when a cell does pinocytosis or phagocytosis or something like that. Extensions of this plasma membrane. First one, let's do microvilli. Microvilli are simply there to increase surface area, primarily for absorption, but perhaps for secretion as well. Microvilli contain only the filaments of the cytoskeletal elements, unlike cilia and flagella, which contain the microtubules of the cytoskeletal elements as well. Cilia are small and hair-like. Flagella are long and tail-like. They have the 9+2 arrangement of the microtubules within their structure. And both of these are involved in movement in some way, either movement of the cell or movement of other things past the cell. And then when we get to histology, we'll see more specifically how that stuff all works. The cytoskeleton is the internal support structure of the cell. Microtubules make it up. Microfilaments and so forth are supporting networks of that for the cytoskeleton. Cytoskeleton is involved in cellular movement in some cases, amoeboid movement like a white blood cell, or the cytoskeleton is also involved in cell division. It forms the spindle apparatus, which separates the chromosomes and the chromatids as appropriate. Lysosomes are kind of like the digestive capsules or vesicles in the cell. Peroxisomes are largely a detox kind of thing. They can help eliminate things like free radicals and whatnot in that process. So all of that is indicated on these two pages. Don't forget also the cytoplasm is the liquid semi-gelatinous interior of the cell. The plasma membrane is the outer barrier. Now on page 70, it talks about the plasma membrane a little bit. Talks about membrane proteins. There's a good picture of the plasma membrane there, give you an idea of what it looks like. Remember, phospholipid bilayer, fluid mosaic model when you incorporate the membrane proteins as well. channels and everything else is indicated. Carrier proteins, a lot of this relates back to transport that we talked about already. Cytosol, cytoplasm, we can use those interchangeably here. Page 72 starts talking about the cytoskeleton. Microtubules are the main part. On page 73, it has some microvilli there showing just a lot of these smaller filaments of the cytoskeleton going into those. 74 and 75 goes into cilia and flagella. They actually have the centrioles there. Remember, centrioles are involved in the cell division process. The 9+2 arrangement of microtubules is there as well. They're described on page 74, cilia and flagella, and 75. Don't get into the details of exactly the molecular machinery that causes cilia and flagellar movement. That's more for a molecular biology class. Just know that they do provide movement. 76, don't worry about proteasomes, as I mentioned. Endoplasmic reticulum starts there. Rough ER and smooth ER. There are pictures on the bottom. There are micrographs on the bottom as well of these pages. Golgi apparatus is there. 78 and 79 is a great double-page presentation of the whole endomembrane system, as well as the processes of transcription and translation. On the left is a nucleus with the DNA in there. You can see messenger RNA coming out, attaching to a ribosome out in the cytoplasm, a protein or peptide being produced. The top there in 3A is free. 3B shows it attached to the rough ER. Protein goes into the lumen, vesicle pinches off, goes to the Golgi apparatus, processing, packaging, etc., etc. Vesicle forms again after that, and some vesicles can fuse with the plasma membrane, spilling the contents to the outside. That's exocytosis. Other vesicles can stay in the cell. Lysosomes, for example, are formed this way as well. So that's a great overview of a lot of different things going on there. The step-by-step process listed from steps 1 through 8 across the top. I'll be talking about transcription and translation in just a little bit, but this is a great place to come to as well for that overview. Page 80, another kind of picture of that, but focusing in on the Golgi and the vesicles that are formed. Skip for a moment to page 83. That is the nucleus that is shown there. The chromatin material, the chromosomes, DNA with the associated proteins are there as well. Continues on to page 84, showing chromosomes, what they look like, a replicated chromosome with the chromatids joined at the centromere. And remember, chromosomes are composed of the DNA double helix along with the histone proteins that accounts for the coiling and everything else that can happen as the cell goes through its cycle. So the last thing I'm mentioning here is the mitochondria. I'm leaving that for last because I want to spend a little time on cellular respiration just to bring out some key points for this. This is something people tend to have some trouble with. So back on page 81, there is the mitochondria, and on the very bottom there is just a diagram of the mitochondria, and that includes the different processes of cellular respiration there as well. It's kind of follow the arrows thing. It's a very simplified form of what you did in Bio 111, and we're really not going to get too much more complicated than this, but I do want to put the key things on the board so you understand the big picture of what is happening. Now, depending on who you had for 111 or where you may have taken the equivalent course if you transferred in from somewhere else, you may or may not have had to memorize all the 10 steps of glycolysis and the enzymes involved, or you may have had to memorize the Krebs cycle and everything that went on there. The same thing with the electron transport chain. You had to know all those proteins that are involved, the cytochromes and all of that. None of that is applicable here. I'm not going to have you memorize all those bunch of things. If you know that, great. If you dumped it after the last exam, that's fine. You may have to recall it if you take microbiology. But for here, we're looking only at the key steps. and what the process of cellular respiration is all about. Bottom line, it is an energy transfer process. We are capturing energy that is stored in glucose molecules. That's where the process begins. And we are converting that energy ultimately and putting it into ATP. That is the goal. So glucose is our starting molecule. There is stored potential chemical energy there in the carbon-carbon bonds. And the entire process of cellular respiration, anaerobic, anaerobic, and all of that, the goal is to make ATP, to take that energy that was here and put it into those bonds between the phosphate groups of ADP and between 2 and 3, phosphates 2 and 3. That's where we want to put that energy so the cell can then use that for metabolic processes. That is what cellular respiration is all about. And yes, it's a lot of steps and everything else along the way, and you can spend your whole life studying various parts of it. But for here, understand the goal, the big picture. And then if you have that, then you can understand some of the minutiae as you would be looking at each individual thing along the way. So to that end, let me start over here, and yep, you can still see over here. So the first part of cellular respiration is glycolysis. Glycolysis occurs in the cytoplasm. It is an anaerobic process. Oxygen is not required for this to take place. And here is where we have stored energy in the carbon-carbon bonds, specifically within the glucose molecule. And we're going to tap into those and put them eventually into ATP. Now, we can't do it all at once. If we did, we would literally have an explosion. We have to tap into it a little at a time. The analogy I use is that of a car with gasoline in the tank and the engine providing power and that kind of a thing. And the engine does not, if you're familiar with how the engine works, the gasoline and air mixture doesn't explode in the cylinders, it's burned. There's a difference between those two. An explosion is an uncontrolled release of energy. That's what would happen if you threw a match into the gas tank. But that's not what the engine does. The engine burns the gasoline. It's a controlled reaction where energy is harvested, which will actually drive the car. Same thing is happening here. The glucose molecule, which we are starting with, has the stored energy, and we are not going to throw a match into it and have it explode. We are going to burn the glucose and then extract the energy from that and simply transfer it to the ATP molecule. So the other thing, well, I think of it to remember about cellular respiration is the fact that almost all of it involves redox reactions. Remember, oxidation reduction reactions. And simply put, redox reactions involve a transfer of energy, which is kind of what we're doing in the whole process here. And when there's many different ways to define it. If you've had some chemistry, you probably had some crazy ways to look at it. For here, for our purposes, for redox reactions, we are following electrons. Electrons equal energy. And if a molecule loses an electron, it is oxidized. It has lost energy. So oxidation is the loss of energy by the loss of electrons. Conversely, a molecule can gain an electron and thus gain energy. That is reduction. Reduction is a gain or an increase in energy by gaining electrons. Okay? So throughout the entire process of cellular respiration, electrons are going to be going back and forth between many different molecules, and that is a transfer of energy. So remember redox reactions. So glucose is what we start with. And then we have the steps of glycolysis going on here. There is the first part, which is the energy investment phase. We have to put energy into this to make it work, and that's in the form of ATP. Then the second part is the energy payoff phase, where we get energy back. And we have a net gain of 2 ATP when this process is all said and done. and we have 2 molecules of pyruvate. So glycolysis is, if you say a glycolysis, you get an idea what's happening. Glyco referring to sugars, in this case glucose. Lysis means to split something. And glucose is a 6-carbon sugar. And right about here at the midpoint, it is split into 2 3-carbon sugars. So we get 2 pyruvates out of this once the process has reached this point. Now, we're not worried about all the minutiae and everything else that's going on here. If you had to learn that previously, part of me says my heart goes out to you. The other part of me says, no, nothing bad about learning that. But either way, we're going to be focusing just on the key steps here. So glucose, investment phase, payoff phase, pyruvate is produced. And we are also going to get 2 other things. Let me write over here. We have a net of 2 ATP are produced, 2 ATP go in, 4 ATP come out, you subtract, and you get 2 ATP that are being produced. We also have an NADH molecule that is, 2 of them actually, that are produced in the process here. Remember NADHs, NAD+, these are the electron shuttles, the electron carriers. These are the main ones that are always discussed when the redox reactions are mentioned. So they're involved in this process, but these are going to be the products of glycolysis. So we have 2 ATP, and with that ATP can be used by the cell for metabolism. Perfect. NADH is an electron shuttle, an energy shuttle. It's going to take energy that it captured from here and take it somewhere else. Hopefully you know where that is, but we're going to talk about it a little bit later. And we have pyruvate. We have 2 of these pyruvate molecules. I'm going to put an asterisk here next to the pyruvate because it is here where the majority of the energy that was in the glucose still exists. We've captured some of it, but the majority, the major portion of the energy is in the pyruvate, and that still needs to be tapped into. And to do that, let's go here. I'm going to figure out where you can see. Okay. Let's see. Pyruvate. So, if oxygen is available, then the pyruvate is going to be further acted upon, and that's the next thing that I'm going to do. So I want to erase this over here. And let me come over to this side where you can still see. That's U. So if oxygen is available, the pyruvate is going to be acted upon a little bit further. We said most of the energy is still here, and the pyruvate is going to be converted to something called acetyl-CoA. This is what is often sometimes called the transition reaction. There are a couple other names for it, but the goal is to produce acetyl-CoA. In this process, another NADH is produced. Also, CO2 is produced by this process. And acetyl-CoA, and we're going to draw kind of a dotted line here. We are now going to be transitioning into the mitochondria. Pyruvate is produced in the cytoplasm as an end product of glycolysis, and during the transition reaction, it is actively transported into the mitochondria and becomes acetyl-CoA. And acetyl-CoA will then enter, I'll put this down here like so, will then enter the Krebs cycle, also known as the citric acid cycle. And the Krebs cycle takes place in the matrix of the mitochondria in the liquid aqueous interior. And it is a cycle. As long as parts are there, it will continue to go round and round. And the key thing about the Krebs cycle is we're going to look at what we are going to obtain from it. Again, energy transfer is going to take place. In glycolysis, we started with glucose, and we took some of that energy, we made some ATP, we made an NADH, and we made the pyruvate. Now we converted it to acetyl-CoA, which is still high energy. And in the Krebs cycle, we're going to extract even more energy out of the molecules as they are produced. So what we get out of the Krebs cycle is we get some more NADH, some more electron shuttles. We also get another electron shuttle, FADH2, and we also get some more ATP. We get technically two more. It is formed into GTP first, which is an analog to ATP and can be converted to it. But this is what comes out of the Krebs cycle. And then, of course, we have to regenerate the molecule, the next one that captures the next acetyl-CoA as it comes in. So the NADH and FADH2 are electron shuttles. They are going to shuttle electrons and energy to the same place that the NADH from glycolysis is going, and the same place that the NADH that was produced here is going to go. One thing about the ATP real quick, and I'm going to write this over here. The ATP that's produced by the Krebs cycle and by glycolysis is produced by substrate level phosphorylation. So just go ahead and write down substrate level phosphorylation. That's the classic enzyme substrate complex, products produced mechanism that was illustrated in the previous chapter. So both glycolysis and the Krebs cycle generate ATP by substrate level phosphorylation. And this ATP can be used by the cell for metabolism. Then the next thing, we're going to take the NADHs and the FADH2, and we will go to the electron transport chain. And you can still see it right over here. Yeah. And in addition, the other NADHs that came from glycolysis and the one that came from here are all going to the electron transport chain. The electron transport chain is a series of proteins that are in the cristae, the inner membrane of the mitochondria. And the goal of the electron transport chain is to produce a proton gradient, to establish an artificial proton, which is a hydrogen ion thing. Let me write this over here. So that is what the electron transport chain does. The electron transport chain does not produce ATP. If anybody told you that, they're wrong. The electron transport chain produces this proton gradient. The energy supplied by the electron shuttles that are here, they come to the electron transport chain and they drop off their electrons to the proteins in the electron transport chain. And as the electrons go from one protein to the next, they discharge energy. Some of the proteins that are in this chain are proton pumps. You learned about the sodium-potassium pump, which pumps potassium and sodium. Proton pumps pump protons. And the energy that the pumps need to establish the artificial gradient is provided by these electrons, which got their energy from either the Krebs cycle or glycolysis, or wherever it came from. Excuse me. And the important thing about the electron transport chain is here is where the oxygen is utilized. Atomic oxygen, not molecular, atomic oxygen is at the quote-unquote bottom of the electron transport chain. And because oxygen is so highly electronegative, it craves electrons, it wants electrons, it almost like a magnet pulls the electrons down the electron transport chain after they're dropped off up here by the electron shuttles. And as the electrons go through, they drive the pumps. The protons are pumped from one side to the other, creating this artificial concentration gradient. Now, when you have a high concentration of something on one side and a low concentration of something on the other side, what wants to happen? It wants to equilibrate. These protons would love to come back through and reach equilibrium. This is an unstable state. We have a high energy situation here, and we would like to lower some of that energy. Now, the protons can come back through, but they can only come through in a couple of places, and that is where the enzyme ATP synthase is located. The enzyme ATP synthase is interspersed along all of these electron transport chains, and the enzyme has a channel, basically a tube, where these protons can come back through. And the analogy I use is that of having a large dam with a gazillion gallons of water backed up, lot of high energy of water on the backside of that dam, and only allowing the water to come through in a few select sluice gates. And when you open those, it shoots through like a jet. And these protons are coming through ATP synthase very, very rapidly, very high speed. And if you remember in the 111 book, it had the structure of ATP synthase. There is this rotor spinning wheel thing that the protons hit, the thing spins around, and that provides the energy for the enzyme to take ADP and join the phosphate to it to make ATP. So the energy that is established in the artificial proton gradient is used to drive the ATP synthase to make the ATP molecule. And this is and the process by which ATP synthase makes ATP is oxidative phosphorylation. And the difference between this and substrate level, in substrate level, you have the molecule containing a phosphate group, and you have adenosine diphosphate ADP. The enzyme simply transfers the phosphate from that other molecule to the ADP, making ATP. In oxidative phosphorylation, you have ADP and inorganic phosphate, just a phosphate group floating around. And the ATP synthase here can take the inorganic phosphate and attach it to the ADP. That is oxidative phosphorylation. And the combination of oxidative phosphorylation with the electron transport chain together is what's known as chemiosmosis. So that is the overview of cellular respiration for what we need to know for our purposes here. And that is, again, summarized on the bottom of page 81. So starting with glucose and forming pyruvate in the cytoplasm, either way, bracketed up there, and then pyruvate going into the citric acid cycle, and then the citric acid cycle proceeding to the electron transport chain. It talks about enzymes and coenzymes. Christie, that's the electron transport chain, and ATP being produced as a consequence of that. So this little picture down there is not all inclusive. They should have had some ATPs coming out of glycolysis and so forth, but this is the big picture that we need to be familiar with. And it is described also on page 82. Down the left-hand side, mostly, don't worry about membrane flow, that's with the plasma membrane. But the left side of 82 kind of summarizes this whole bit about cellular respiration. So one final thing I do want to mention here, because it will undoubtedly come up at some point, that is fermentation. So aerobic respiration occurs when oxygen is available to be the electron acceptor at the electron transport chain. Anaerobic respiration means, yes, it's respiration without oxygen, but anaerobic respiration and fermentation are not the same thing. Anaerobic respiration also will use an electron transport chain, but it uses something other than oxygen as the final acceptor. It could be sulfur, it could be several other molecules. A lot of bacteria and things will do anaerobic respiration. In micro, you'll learn about that. Fermentation is a completely different process. Aerobic respiration and anaerobic respiration both produce ATP. Fermentation does not. Fermentation, the goal of fermentation is to regenerate NAD+. That is what the goal of fermentation is, to regenerate this, because NAD plus is important in the initiating the payoff phase of glycolysis. Without this, glycolysis would stop. So fermentation essentially keeps glycolysis going. It takes pyruvate and takes energy from that, and it will oxidize the NADH to NAD plus, and in animals, humans, it will convert the pyruvate, excuse me, into lactate or lactic acid. We'll talk about that a little bit more again in chapter 10 on muscle physiology, where some of you may be familiar with, a lot of activity generates lactic acid in the blood, et cetera, and that's the fermentation process that is taking place. So again, more on that when we get to chapter 10. So that covers the cellular respiration thing. The next thing I want to mention quick is protein synthesis, transcription, and translation. And this starts on page 85. So if you go to the bottom left of page 85, and then over to the right, it talks about transcription first, and then it talks about translation back on next on page 86 and into 87. And on the bottom of page 86, there is the process of transcription. Now, a little more detail there than we really need to worry about. For our purposes, and I'll summarize this real quick, transcription happens in the nucleus, translation happens in the cytoplasm. Transcription is going from DNA to RNA. In terms of an analogy, you're keeping the same language. When you're transcribing something, you're keeping the same language, you're just putting it in a different form. DNA is nucleotide language. RNA is also nucleotide language, it's just a slightly different form. So that is what is happening in the nucleus. And the goal of transcription is to generate a messenger RNA molecule, a copy of the gene or allele that you're interested in expressing, ultimately. This includes this also relates to things with the genetic code. Know that genetic code is the triplet sequence of nucleotides within a stretch of DNA called the gene or the allele, and each of those triplets is known as a codon. There is a start codon, which is the capital letter. There are 3 stop codons. You don't have to memorize them, but there are 3 stop codons, which are the end of the gene. When a messenger RNA is produced, the initial primary transcript is the exact copy of the gene that was used as the template, and it is then modified through RNA processing or alternative RNA splicing. They're slightly different, but they're about the same for us. And the messenger RNA is shortened. There are introns and exons. Introns are removed. Exons are spliced together. There's a 5' cap and a poly A tail, and then the messenger RNA goes out into the cytoplasm. So that is the process of transcription. There is a little thing about the genetic code on the top of page 87. An example, nothing you need to memorize. You just need to know it's the triplet sequence, the codons that make up the genetic code. And also, almost all of these things are going to be related to RNA, but I'm glad in this book that they also included the triplet code for DNA, because a lot of people forget that DNA has the genetic code, at least initially. So that is as much as you need to know for transcription. Translation is the next thing, and it starts on the bottom of 87, and then 88 and 89 is a nice double-page overview of the process of translation. And translation is exactly that. You are translating something. You're going from one language to a second language. And in cells, the messenger RNA is nucleotide language, but we need to go to protein language. And when you're going to go from one language to another, a very important component that you must have is a translator. And in protein synthesis, the translator is the tRNA. T stands for transfer, not translate, but it's kind of close. But that is the translating molecule. So a transfer RNA, a tRNA, has nucleotide language through most of its structure, but on one end it has a triplet sequence known as the anticodon. That is complementary to the codon. At the opposite end of the tRNA molecule is the correct amino acid for that particular anticodon. Don't worry about how it gets matched. That's not a big deal for us here, but know that it carries that. So that is the translating molecule. So for translation, you need the tRNA, you need the mature messenger RNA, which has come out of the nucleus. You need rRNA, which is ribosomal RNA, because that is part of the enzymatic complex that's going to actually make the protein, which is the ribosome. And the ribosomes can be either free in the cytoplasm or they can be attached to, the rough ER. Either way, the function of the ribosome is the same thing. Now, in translation, like in transcription, there is the initiation step, the elongation step, and the termination step. So on page 88, this is here on the left, is the initiation step, all the way to step number 1 there. And it's the small ribosomal subunit. The first transfer RNA with the correct start anticodon binds to there. The messenger RNA is attached in there, and then the big subunit sandwiches on top of it. The large subunit contains 3 active areas or chambers, sites, whatever you want to call them, E, P, and A. E is the exit, P is the peptide, and A is the amino acid. They may have slightly different names, but that's what that refers to. And that's in figure 2, where the thing is sandwiched in there. And the available codon, which is on the one side in the A site, is now open. The first one is where the start codon was. Now the next one is available. Then the correct amino acid now binds to that, and then the peptide bond forms between the 2 amino acids that are joined together. And the amino acid that was on the middle transfer RNA that's in the center site gets moved over to the one that just came in. And then the entire ribosome will now translocate over one codon, and that moves the amino acids over in this direction. The one that was in the P site in the middle, which is empty, is now in the E site. The one that came in the A site that has the growing peptide chain on top of it is now in the center. The one in the E site gets kicked off, and then the A site is now open. The next amino transfer RNA comes in, peptide bond forms, translocation, the empty tRNA is kicked out, next one comes in, lather, rinse, repeat over and over again. That is steps 2 through 4 as we go along into page 89. And then finally, the last one is the termination step. When the stop codon is reached, something called a release factor comes in and essentially breaks everything up. And then the process of translation is finished. Now, after this, there is more things to do, which we're not going to get into as far as the protein is concerned, but that's transcription and translation. Also be aware of polyribosomes, polysomes, which are multiple ribosomes on the same messenger RNA as it is being translated. So that's all we need to worry about for that. Don't worry about things that can interfere with translation. Don't worry about any regulation of this stuff. I know you did that in 111, or you should have, and that can get a little complicated. But we are just concerned with knowing transcription, it's happening, and know the basic steps in the process. Now, we talked about solutions and everything already, so at this point, we can skip over to page 102, and here we start talking about the cell cycle. And the cell cycle includes interphase and the dividing phases. Remember the 2 main ones, interphase, which is the non-dividing phase, and mitosis or meiosis, which are the dividing phases. Interphase is divided into 3 subphases, 2G or growth phases and an S phase. G1 is the first of these, and this occurs after cell division has been completed. G technically stands for gap, but growth is largely what happens here. So in G1, the cell is growing, it's getting larger, it's making more organelles, it is maturing, whatever it needs to do to get to up to some point where it could potentially divide again. If it is not going to divide again, then it will stay in G1, and this is part of its G0 phase. G0, remember, if you haven't heard this, is a measure of a cell's specialization, its job, what it does for a living. Is it a muscle cell? Is it a nerve cell? Is it a skin cell? Is it any other of the different cells that we have in the body? And depending on what kind of cell it is, what its profession is overall, will determine how frequently it divides, if at all. For example, nerve cells and muscle cells, once they reach a certain level of maturity, never divide again. Skin cells, on the other hand, we have a layer of stem cells under our skin which are constantly dividing and replacing the ones that are being lost above. Same thing with a lot of linings and things on the inside of our body and so forth. Red blood cells, for example, in the bone marrow, there are stem cells in there which are constantly dividing and producing the number of blood cells that we have. So depending on its differentiation or specialization, that's something that's at the very end of this chapter. I'll point at it again once we get there. But do be familiar with the term differentiation and specialization, because that is what leads to a cell's G0. Okay? Whatever the cell's G0 is determines how long a cell's G1 phase is. Now, if a cell is going to divide again, then it enters the S phase, or I should say potentially divide. During the S phase, DNA replication occurs. We have to copy the genetic material so each daughter cell gets an identical complement of what the parent cell had. Now, DNA replication, flip over to page 106. Just 106. That is where it is showing the process of DNA replication. It's a big summary thing all in one page there. But you should be familiar with the steps involved in DNA replication. And conceptually, of course, each strand of the DNA helix is used as a template to generate the new daughter strands that are produced. And here again is where the 5' 3' comes into play in the synthesis direction that takes place. So know the enzymes that are also involved in each of these individual steps. Helicase, opening up the strands, breaking the hydrogen bonds. Single-strand binding proteins, blocks, keeping them apart. Topoisomerase ahead of helicase, breaking and recoiling and relieving the stress of the unwinding of the DNA helix. Primase, producing the RNA primers. DNA polymerase 3, making the first extensions of DNA based from the RNA primers. And then DNA polymerase 1, replacing the RNA primers with DNA, and then DNA ligase filling in any gaps that might remain. Remember origin of replication, remember leading strand and lagging strand, the Okazaki fragments, be familiar with that stuff. Again, not at a super detailed level, but you should know the sequence of things and what's involved in each one. The one in your page in your book here, I think, leaves out a few things, but go back to your Bio 111 notes as you need to. Now, once DNA replication is almost done, and the cell is then still going to divide, then we enter the G2 phase, which is an increased growth, energy storage, producing some extra organelles. Each daughter cell needs to have a minimum complement of organelles and things in order to survive. There are checkpoints within the cell cycle to make sure that things happen as they should. If any of these checkpoints are failed, first, the cell can try to fix the problem. If it can, great, it keeps going. If it can't fix the problem, remember apoptosis, cell death. And in a multicellular organism like ourselves, we can't have cells going rogue. If a cell is going to be, do its own way and go on its own cycle, it's not going to be beneficial for the entire organism. So that cell is sacrificed in favor of all the others that are doing what they should, when they should, and so forth. So apoptosis will take care of any cell most of the time that is not doing its thing. Then, after G2, we begin the actual division phase. Now, mitosis is the division of the nucleus. It is the separation of the chromosomes of the genetic material. During the S phase, when the DNA is copied, the chromosomes are copied, and the two copies are called chromatids. They're held together at the centromere. Remember, the number of chromosomes, the diploid number, does not change. Humans have 46 chromosomes. When DNA replication is completed, we still have 46 chromosomes. We just have an extra copy of every one. The quantity of DNA has doubled, that is true, but the number of chromosomes has still stayed the same. So that when cell division is complete, each daughter cell now has 46 chromosomes once again. Meiosis, of course, is a separate scenario. We'll mention that real quick once we get done with this. So mitosis is nuclear division. And there are 4 or 5 steps, depending on what book you're looking at, in mitosis. The first is prophase, where what I will do is leave it to you to look in your book. It's on page 104 and 105, 105 primarily, which is going to show the different phases of mitosis, and underneath at the bottom are visual cues and what's happening in each of these particular cases. I'll leave you to look at that. So your book talks about early prophase and late prophase. That is just one way to look at it. The Bio 111 book and others talk about prophase and pro-metaphase. So prophase, because it's so long and because different things happen in each portion of it, has been subdivided into 2 subphases. Prophase and early prophase are basically the same. Late prophase and pro-metaphase are the same. So some of you may have to switch your terminology a little bit here, but I'm just going by what your book has. Then metaphase is the next one, where the chromosomes are all lined up along the equatorial plate. After that is anaphase, where the chromatids are separating, and I'm emphasizing chromatids because we're talking about mitosis here. Anaphase is the shortest phase. And remember, the cytoskeleton forms the spindle, which pulls the chromatids apart. After anaphase is telophase, and telophase is the reverse of prophase. No more complicated than that, except now we got 2 potential cells which are in the process of forming. So we have separated the chromatids, the chromosomes. We have made 2 nuclei which are reforming, but we still have 1 cell, at least temporarily. Now that is starting to also separate. As telophase is underway, cytokinesis, which is the actual division of the cell, is beginning and going on. Now, since we're dealing with animal cells, cytokinesis involves a cleavage furrow, or they're calling it a contractile ring sometimes. Basically, the equatorial region pinches together and then the cells separate out like that. Okay, very straightforward. That is the last step that is there on page 105. The rest of the cell cycle is mentioned on page 104. There is even the G0 phases mentioned there at the very bottom as well. So you can look at it's a pie chart kind of thing, so you can look at that. Don't worry about the times that are listed there. Again, those are averages. I'm not going to test you on any of these times, but know the phases and the things that happen during each one of those. One thing your book doesn't mention, at least not here, is meiosis. It mentions it in the chapter on reproduction, which is at the end of 202. But don't forget about meiosis. Meiosis is the first half of sexual reproduction. Fertilization is the second part. And meiosis is a specialized form of mitosis. Meiosis reduces the chromosome number to haploid, so the mature cells that are produced, the gametes, will have now 23 chromosomes instead of 46. Mitosis produces 2 genetically identical daughter cells. Cloning is basically what's happening. Meiosis produces 4 unique haploid cells, the gametes, sperm and egg in male and female as appropriate. The key thing in meiosis that makes it different happens during prophase, and it is the homologous pairing of chromosomes, or the pairing of the homologous chromosomes. Remember Mendelian genetics, alleles, dominant, recessive, that kind of a thing. The homologous chromosomes contain the respective alleles for the genes that are involved. And in meiosis, the homologs pair up, and those are the ones that are along the equatorial plane during meiosis 1, the first half of it. So that is the key thing, the pairing of homologs. From that, we get crossing over, the recombination that occurs. That increases genetic variation and diversity to help adapt to changing environments. And separately from that, independent assortment, the random arrangement and alignment of the paired homologs at the equatorial plane. So all those things contribute to the variation, which is the goal of meiosis, to incorporate this increased variation, it's a survival benefit, to individuals and the population species as a whole. All of that happens during meiosis 1. Meiosis 1 produces 2 daughter cells that already are haploid, and they are unique with respect to each other. At metaphase 1, it's the chromosomes that separate. The homologous pairs form these things called tetrads. In meiosis 1, the chromosomes separate. Then there is a time called interkinesis, where it's kind of like the cell cycle, it's like an interphase. The exception is there's no S phase. There's no further DNA replication. It happened once at the beginning. It's not going to happen again. Then meiosis 2 is essentially identical to mitosis, where in the chromatids now separate, and we get a total of 4 unique haploid daughter cells at the end of this. All right, now in your book, on the bottom, 107, for example, you only need to look at the left-hand side of it, where it completes the discussion of DNA replication. It even talks about telomeres there as well. The right side of 107 talks about abnormal cell growth, cancers. We're not going to worry about that here. There's a table on the bottom of page 107. You can ignore that unless you want to look at it, but nothing I'm going to test you on. 108 you can also essentially ignore as well. Do look at the bottom of 108, the clinical note. There it talks about telomerase, which is the enzyme that restores the telomeres in DNA replication. Don't worry so much about the aging and cancer, but to reference the telomerase thing. And on page 109, section 3-11, talks about cellular differentiation. The word is even in bold there in the text itself. Cellular differentiation, specialization. Those two are together. That is what is going to be relevant to the G0 phase of the cell cycle, as was mentioned earlier. So that covers the Bio 111 review. Yep, there was a lot of material that you learned in the whole semester, and I condensed it down to 3 more or less 1-hour lectures. But it is stuff that you need to be familiar with. The majority of the exam 1 is going to be on a lot of this material. Also remember, chapter 1 in the book is also going to be on exam 1. Organ systems, some very, very basic terminology type stuff. The section about the body regions, the directional terms, that's all for the terminology test. So that is exercise 1 in the lab manual. There'll be only a handful of things on exam 1 regarding terminology. I might ask, what does distal mean, or what's the acromial region? Just a very few things. The bulk of the terminology is the terminology test, which is a lab thing. But don't ignore it completely, because chapter 1 does have some things relevant to that as well. So study for the terminology thing, the vocabulary, that's the terminology test. A few questions will relate to it on exam 1, but the bulk of exam 1 is organ systems and this Bio 111 review material.